Hi everyone! This is a recording of the Chromatin and histones lecture. Good job everybody on the DNA technologies quiz. You guys did quite well and I hope you guys continue to do better in the quizzes. So chromatin and histones and as well as genomes are the two topics I'm hoping to cover this week, as well as on Friday you guys will need to turn in your homework number 1. The file is available for download on the Wire Courses Files section. And then between 6 and 8 p.m. we are going to have our exams through the Wire Courses shell, similarly to the quiz. I have posted an announcement that hopefully everybody has seen with both my email and my phone number in case some emergencies happen and you guys are unable to complete the exam. If you cannot make that time between 6 and 8 p.m., please email me as soon as possible so I can figure out if I could do alternative arrangements. Now, chromatin and histones. It is a topic that has unlikely been covered in your standard biology classes. This is a little bit more of a molecular biology subject than, say, DNA replication, which is a very generic biological topic. So hopefully you guys get to learn a little bit more today than or at least get exposed to a topic that is new to you. So how much DNA? You guys already have seen this slide when we talked about DNA replication. The fact that each cell in our body has 2.2 meters or about 3 yards of DNA in every nucleus is amazing. So this is an electron microscope photo of a typical eukaryotic cell, and you can see that the nucleus takes about 10% of the cell, yet it's capable of fitting 2.2 meters of the DNA molecules. That's an equivalent of 24 miles of very thin thread fitted inside a tennis ball. So how can all this DNA fit in? Well, the answer, of course, is very careful packaging. The DNA is spooled and compacted in many ways before it can be fit into the nucleus. And then it's even compacted further to create these distinct X-shaped chromosomes that you guys, the picture that you guys associate the word chromosome with. So that's highly condensed chromatin that you need to have in order to carefully divide DNA during cell division. But DNA is there's not just the problem of DNA, the size of DNA molecules inside our nuclei. It's also the fact that we express a lot of genes from these DNA molecules at any given time. A single cell is likely to express about 1,800 genes at any given time inside of itself. So the DNA cannot just be highly compacted to fit into the nucleus. It also has to be in some way accessible so that mRNA transcription machinery can go into the different regions of the chromatin and copy the correct instructions to be made into protein. So how is that done? Well, the carefully packed DNA is also very carefully cataloged and marked, and the different regions of the DNA are set aside either from gene transcription or for gene silencing, preventing mRNA production from that area. And all of this has to, of course, be tightly regulated. So how's this done? It's of course done with proteins. It's the proteins who catalog and mark the DNA in various ways. And this combination of proteins and DNA that make up the chromosomes is called chromatin. And here I would like to make a important distinction. The structure that you guys see here, this black fuzzy X, is a chromosome. However, it is made up of chromatin. That's the substance, the material that the chromosomes are made out of. And chromatin is a mixture of DNA molecule and protein. And here there's a typical mitotic chromosome. Here will have two sister chromatids, a centromere that's holding them together, and then telomeres on both ends. Hopefully you guys remember that from basic biology. So as you just saw chromosomes, especially pre-division chromosomes, are highly compacted combinations of protein and DNA. Now, a single DNA molecule that is not about to be divided will not be all uniformly compacted like a mitotic chromosome, but will have different areas with different levels of condensation or compaction or packaging. So it can be divided. Generally, you can speak about heterochromatin, which is a highly compacted chromatin that usually have low gene density found in it. There's very few genes inside that chromatin, and those areas of chromatin will have very low gene expression because they're so compacted, and it's very hard for the RNA polymerases to get in there. And then the other term is euchromatin, those are the areas of the chromosome that includes most genes, especially important housekeeping genes that make sure that the cell works correctly. And those areas can have variable gene expression, but it's not as highly compacted as heterochromatin is. And of course, the chromatin compaction state is highly regulated, and the level of compaction of the chromatin will have a strong effect on whether a certain gene can be converted into mRNA or not, aka can the gene be expressed or not. And here's the picture. So there's a picture of your typical chromosome. Now I put it on its side. Let's pretend it's not as compacted as it is here. And of course, every chromosome will have a telomere, two telomeres, and a centromere. And you guys can see it here. Both centromeres and telomeres are regions of highly compacted heterochromatin and usually host very few genes. And then other areas of the chromosome will have regions of less compacted chromatin called euchromatin, and then also some areas are still going to be compacted, such as heterochromatin. And genes can be found sometimes in heterochromatin, but usually in euchromatin areas, especially genes that are generally expressed by all cells. So how is this organization occurring? How are the proteins organizing the DNA molecule to create euchromatin and heterochromatin areas? So the experiments that I'm going to describe right now were done back in the 60s and 50s, shortly after DNA structure was understood. And one of the experiments done by Roger Kornberg was to look at how accessible the proper chromatin material was to nucleases. Now, if you guys remember, the word nuclease indicates a protein, specifically an enzyme, that can take a DNA molecule polymer and break it down into the nucleotide monomers. Remember how the DNA polymerase had an exonuclease activity where instead of building the DNA polymer, it was now taking off incorrect bases from it. So nuclease means it cuts up DNA. So the experiment that they did was to carefully isolate chromatin, aka not just DNA molecule, but also the proteins that were attached to the DNA molecule, and then digest it, incubate it together with a nuclease that would only cleave where the DNA was not protected by protein. So if there was very little protein protecting the DNA, the nuclease would simply destroy the DNA, and you would get just a really bright band at very, very, very low molecular weight, and it would be kind of random because a nuclease does not have any specific places it likes to cut. It just cleaves the phosphodiester bonds. But what was observed after the nuclease digested chromatin was run out, was separated by size using gel electrophoresis. Remember that it will separate DNA molecules based on size, they observed banding, which suggested, and it was banding in increments of 200 base pairs. So the smallest band was 200 base pairs. Then they also saw 400 base pair bands, 600, 800, etc. You can see that this banding pattern continues for a good long time. But they didn't see any bands less than 200 base pair band. And that suggested to the Roger Kornberg and his group that the chromatin, aka DNA and protein, was existed together in these 200 base pair chunks. The 200 base pair chunks of a DNA molecule were very well protected by the proteins that were coating the DNA. And about in the same time, other scientists were also looking at chromatin, aka the DNA and protein complexes, not just pure DNA, under an electron microscope. So, and what they observed were these beads on a string appearance that you guys see here. These little beads, and then they're connected with uncoated DNA, and there's another bead, and so forth. And so it was when these groups communicated, it was quickly realized that the 200 base pair chunks seen here were most likely, based on measurements, were these beads on a string observed in electron micrographs. So what are these beads? They are called nucleosomes. So it takes about 147 nucleate base pairs of DNA plus the important proteins that create a nucleosome to create these chunks. The reason Roger Kornberg group was getting 200 base pairs instead of 147 is because even that way the nucleases couldn't quite get in, and they would be cutting somewhere in the middle. And nucleosomes are DNA plus protein, and nucleosomes can be arranged on DNA in various ways. When there's a lot of open uncoated DNA in between nucleosomes, it is an area of euchromatin where highly expressed genes are present. And if these nucleosomes have been packaged very, very densely, actually far more densely than you see here, is the area of heterochromatin where little gene expression can occur. So what are nucleosomes? So they are DNA molecule plus 8 proteins. So there's 4 kinds of proteins inside the nucleosomes, and then they double up, creating 8 protein structure. So this octameric histonic core is proteins, and then 147 base pairs is the DNA that has been spooled onto this little spool. And if you break up this octameric histone core, so the proteins are called histones. Each single one of these things is called a histone. This is histone H4, H3, histone H2A, histone 2B, histone 4, histone 3, histone 2A, and histone 2B, H being the histone. And if you break them apart, you'll realize that each of these spools contains 8 different proteins, 8 protein units, and there will be 2 histone 2As, 2 histone 2Bs, and then a tetramer of histone 3 and histone 4 monomers, which make up this tetrameric subunit. And if you take these proteins and you run it on a gel, so you can run proteins on a gel the same way as you can run DNA, not because proteins are all negatively charged, but because if you put them into a very high electric field, they do develop a charge. And so they can be separated. And you guys can see here that they're all about the same size. There's 2 A's and 2 B's, very similar. H3 is slightly bigger than H4, so faster things move. Faster things are smaller, slower things are larger. And then there's H1 protein we're actually not going to discuss in class because it is not part of the histone spool, but it associates with the spool and helps convert euchromatin into heterochromatin. There's a lot more H1 proteins inside heterochromatin nucleosomes. But it's a little bit more than I'd like to cover in this class. So, Nucleosomes are DNA plus histone proteins, of which there is 8 and there's 4 kinds: 2A, 2B, 3, and 4. So let's look more at histones. So all eukaryotic DNA is spooled on these histones. They're highly conserved and they're very, very ancient little proteins. Every single eukaryote known will have histones. And they, as I said, are made up of 8 histone proteins, 2 molecules each of 2A, 2B, 3, and 4. And again, this is how they look. The beads in a string appearance is exactly how the nucleosomes look like. And the core of each nucleosome is a histone octamer. And this is the actual crystal structure of a typical nucleosome. So one important thing that I want to talk to you about in this slide and in the next one is that histones are positively charged. They have a lot of arginines and lysine amino acids inside of them, which are basic positively charged amino acids. And that makes sense because that's how you interact with DNA. Oh yeah, and one more thing to point out is when the DNA is bound to a histone, it's a nucleosome. If it's just the proteins, they're histones. Okay, so here's a lecture question that I think you are totally able to answer based on the information I just gave you. So pause this lecture and think about how to answer this question.